The overall goal of the following experiment is to quantify nutrients in pollen and bee bread made by two honeybee subspecies and measure consumption of protein titers. This is achieved by presenting European and African bees with the same pollen source in a confined area and allowing the bees to store the pollen in their colonies as bee bread. Next, the bee bread is fed to newly emerged European and African bees in cages. Then, bees in the cages are sampled after 4, 7, and 11 days of feeding, and the amount of bee bread remaining in each cage is measured to estimate consumption. Finally, the concentration of nutrients, including protein and amino acids in the pollen and bee bread made by European and Africanized bees, are measured and compared between the races of bees. The results show that bee bread differs from pollen in protein and some amino acid concentrations, as well as Hemolymph protein titers.
This method can help answer key questions in studies of honeybee nutrition, such as the role of pollen source on the nutrients in bee bread, and how those nutrients are acquired by bees of different subspecies, races, and genotypes. Demonstrating the techniques is Emily Watkins de Jong, who is a student technician in my laboratory.
To begin, after placing pollen traps on honeybee colonies, collect the pollen. Use a coffee grinder to grind the pollen into a fine powder. Establish 5 colonies each of African honeybees, or AHB, and European honeybees, or EHB, in an enclosed flight area, or